This is the second video with regard to genomic testing by paediatricians. In this video, we will consider genomic testing and consent. In the previous video, Oliver, aged 42 months, remains non-verbal, has a sleep disorder and now has had recent onset of seizures. The family now is in a better space to consider genomic testing. In this video, we will consider learning outcome number 2, which genomic test to order, obtaining consent, organising the genomic testing on a patient. At any time during this process, referral or discussion with your local genetic service is possible and advised. Let's consider which genomic test to order. Just to remind you, we have 3 billion units, that is base pairs, of DNA, which are made up of 4 specific proteins. These are collectively termed DNA. They are in a specific sequence within a structure called a gene. We have 20,000 genes that sit like beads on a string on our 23 pairs of chromosomes. These are housed in the nucleus of every cell of our trillions of cells. The sequence of a gene is like an instruction manual or recipe, coding for a specific protein that is involved in the development and function of our body. Genetic testing can either be done with a single gene via Sanger sequencing or genomic testing, that is testing all genes. This may be with a gene panel or whole exome sequencing, the patient only, called a singleton, or a patient and both biological parents, which is called a trio, or a whole genome sequencing, which can also be done on a singleton or a trio. To explain the differences between whole exome and whole genome sequencing, it is important to remember that our DNA is divided into introns and exons. Exons are the protein coding parts of our DNA and make up 2% of all our genomic material. Introns are the non-coding parts of our DNA. Therefore, sequencing can be done of the whole genome, that is, both our introns and our exons. Alternatively, we can sequence the whole exome, which is our 20,000 genes. However, it is only the protein coding parts, that is, the exons of our genomic material. Of the 20,000 genes, approximately 5,000 genes can be related back to human disease. This is termed the clinical exome. The clinical exome is expanding all the time with gene discovery as more genes are identified and related back to human disease. So what is a gene panel? When we do genomic testing, we can sequence the whole exome, but only examine the part of the exome that is related to the clinical presentation. For example, with Noonan syndrome, there are about 20 causative genes known to date. When we sequence the whole exome, we may only choose to examine those genes that are associated with Noonan syndrome. This is termed a gene panel. If this is negative, because we have sequenced the whole exome previously, we can revisit as time goes by and reanalyze either all the genes associated with human disease or alternatively look at more Noonan syndrome genes as they are described with time. So choosing which genomic test depends on the clinical presentation. We may only choose to sequence one gene. For example, in achondroplasia, the only gene causative of this condition is FGFR3, and we may request Sanger sequencing. However, other clinical presentations are associated with multiple genes. For example, Noonan syndrome, Marfan syndrome, and hearing loss. We would therefore choose a gene panel. However, other clinical presentations are much less gene-specific and are associated with hundreds and hundreds of genes, such as intellectual disability, multiple congenital anomalies, or severe epilepsy in association with intellectual disability. In this situation, we would consider whole exome sequencing, in other words, examining all our protein coding genes. In terms of syndromes and intellectual disability, which will be the common reason you order the test, Whole exome sequencing is a test of choice because of yield. There is an up to 41% detection of a causative gene from genomic testing for intellectual disability. Whole exome is better than the gene panel for intellectual disabilities, and trios are better than singleton, as they have a 10 to 15% higher yield. In addition, there is a lower rate of variance of uncertain significance as parental sequencing is used as a reference genome to discard normal variations. If selecting a trio, considerations include: are both parents available? And concerns regarding insurance applications, which will be explored later in the next video. Let's get back to Oliver. We are now going to organise whole exome sequencing. We consult clinical genetics, and it is arranged that we do both biological parents, that is a trio test, as well as Oliver. This will require obtaining consent. Let's explore Learning Outcome 2, Part B, Obtaining Consent. A genetic or genomic test has implications for the family, so obtaining consent is an important part of the process. Obtaining consent means filling in a consent form for each person undergoing genomic or genetic testing. In other words, for a trio test, a consent will be required for the child and one each for his or her parents. Currently, New South Wales Health has a 2-page genomic testing consent form. These consent forms may be accessed at local genetic services. Contact details for these genetic services are at www.genetics.edu.au. There will be a national consent form in the future. Informed consent requires a quick summary of genetics for patients and their families. In order to explain genomic testing, the analogy of the library may be used. DNA is like letters made up of 4 chemical units that make up words in the long continuous sentence of a gene. The genes are like instructions or recipe books. Humans have 20,000 books on 23 pairs of shelves in our library. To date, we understand the meaning of 5,000 of these books, but this is increasing all the time. Genomic testing is checking for spelling changes in these books. We can either examine all the books, whole exome or whole genome testing, or we alternatively can select a set of books, that is a gene panel, as you would for Noonan syndrome, or we can only look at one book, Sanger sequencing, achondroplasia or cystic fibrosis. When explaining consent to families, it is important to remember that we may be sequencing 20,000 genes, but only 5,000 to date have been related back to clinical disease. Gene discovery is occurring exponentially, and this number will increase with time. The second point to explain is the possible outcomes of genomic test results. Firstly, a genetic cause may be identified. This means that a likely pathogenic variant in a known disease gene has been found. If a genetic diagnosis is identified, this diagnosis may be unique and rare with little natural history information for the child in the future. In addition, genetic diagnoses may have implications in terms of decreased lifespan or degeneration. The second outcome is possibly a variant of uncertain significance. In other words, a change in a gene that we cannot understand at the present time, an unclear or maybe answer. The third possible outcome may be a negative result. To continue points to remember, incidental findings may arise. They are rare at 1 to 2%. However, it's important to point out to families that we are only examining child's data and not actively looking at parental data. Parental data is only examined if incidental findings are found in the child. Different labs have different screening for incidental findings. It is important to check with your lab what approach they have to incidental findings. Secondly, insurance implications mean there is no effect on health insurance, but new applications for life and income protection insurance may mean that declaration of these incidental findings is mandatory. The third issue is in terms of whether a singleton or a trio is selected. There are less variants of uncertain significance found in a trio as parental data is available for comparison. However, there is a higher risk of incidental findings, as genes unrelated to the clinical presentation are examined. Fourthly, unexpected family relationships may be revealed, such as non-paternity. If you need some more tips to give you confidence when obtaining genomic consent, there is more for you at the Centre for Genetics Education website, and links will be provided on the resources page at the completion of this course. Part C, Organising Genomic Testing on a Child. So, how do you organise the genomic test? Which lab to send your genomic test to? Please contact your local genetic service for advice about which lab to order genomic testing from. This will depend on your location. The current New South Wales laboratories offering genomic testing are Randwick Genomics Laboratory and the Molecular Genetics Laboratory at Children's Hospital Westmead. Testing must be done by an accredited Australian laboratory. In terms of selecting the lab, it would be important to discuss with your local genetics service about which would be the most appropriate. For example, the issue regarding generic versus specialised testing expertise. It is important to note that testing at international labs will not be funded through Medicare. The following documentation is required when referring exome testing. 1. Referral form for genetic testing signed by the referrer. 2. Phenotype information on request form or separate attached letter with detailed clinical information. Each laboratory will have their own requirements. Please consult with clinical genetics to check this. 3. Genetic consent forms signed by the patient, and in the case of trio exome, by both biological parents as well. The request and phenotype form is dependent on the laboratory selected. Most laboratories require 2 to 10 mL of EDTA blood for DNA extraction and storage. In the request form, it is important to fulfill Medicare requirements in the clinical information with dysmorphic features and/or one or more major structural congenital anomaly or intellectual disability or global developmental delay of at least moderate severity. It is also important to remember that if a trio is selected, separate request forms are needed for the child and each biological parent. Once the blood and DNA is stored at your chosen lab, send the request form with copy or copies of the consent with detailed clinical information. At this stage, each laboratory will have their own clinical information or phenotype form that will require to be filled in. Please consult with your local genetic service for details of this form. This completes learning outcome number 2. The next video will cover learning outcomes 3 and 4, explaining a genomic test result and knowing when to contact genetic services for assistance.